Module 2, PFK activity in Alzheimer's patients. Bigl and al. Wanted to see the effects that AD had on PFK isoenzyme patterns in the brain, as well as PFK gene expression levels in disease patients. The experimental setup they proposed was extracting brain samples from AD patients as well as non-AD patients. Samples were collected from the hippocampus, occipital cortex, cerebellum, and the frontal cortex. The PFK mRNA was extracted and quantified using reverse transcriptase PCR, as well as ribonuclease protection assay. The results were separated into two graphs. The first one showed the results of the RPA, and the second one showed the results of the reverse transcriptase PCR. The white bars showed the normal samples, and then the black bars showed the AD samples. Note that both the quantification methods showed similar results. The results found that the total PFK levels are elevated in AD patients' hippocampus and frontal cortex. From the quantification information, they devised a percent distribution graph for all three PFK isozymes. They reported there was no significant difference between the isozyme patterns in terms of disease and non-disease samples. So Bigl and al. Found that there was an increased gene expression of all PFK isozymes, but the expression pattern of the isozyme is unchanged between AD and non-AD patients.
Next, Bigl and colleagues addressed the question of whether the formation and deposition of beta-amyloid plaques, another hallmark of Alzheimer's disease, is related to impairment of glucose metabolism in the brains of Alzheimer's patients. Transgenic mice that exhibited progressive age-related deposition of beta-amyloid plaques in their brains due to the overexpression of the Swedish mutation of the human amyloid precursor protein provided an appropriate model to determine the effect of beta-amyloid plaques on the expression and activity of PFK. cPFK mRNA levels and activity were found to be significantly lower in the brains of age-transgenic mice compared to non-transgenic mice that had a high accumulation of beta-amyloid plaques. Correspondingly, the activity of PFK in the cortex of age-transgenic mice was lower than in non-transgenic mice. From the results, it was concluded that a reduction in the expression and activity of PFK and the corresponding reduction in glucose metabolism in age-transgenic mice required the presence of a prolonged and high load of beta-amyloid plaques. mRNA expression patterns of cPFK in brain sections of age-transgenic mice were compared to the locations of beta-amyloid plaques and reactive astrocytes that are produced in the brain in response to neuronal damage. In panels A and C, the cPFK mRNA levels or expression patterns are shown. The beta-amyloid plaques in the same brain section as A are seen as green fluorescence in panel B, and astrocytes in the same brain section as C are indicated by asterisks in panel D. Evidently, cPFK expression was decreased in neurons associated with plaques, but increased in astrocytes distal to the plaques. From this experiment, it was concluded that there are cell-specific changes in PFK expression in the presence of beta-amyloid plaques. In plaque-associated neurons, PFK expression is decreased, and in astrocytes, it is increased. Together, they combine for a net change in PFK activity and expression. If the increase in astrocytes is greater than the decrease in plaque-associated neurons, then PFK activity is seen to increase overall, as is found in human Alzheimer's patients. PFK activity in the age-transgenic mice was found to have decreased overall, which is different than what was found in humans. This was attributed to a lower production of astrocytes in mice than in humans due to milder local neuroinflammation in the mice. The conclusion from this research was that the overall reduction in glucose metabolism observed in the brains of age-transgenic mice with prolonged and high loads of beta-amyloid plaques occurs as a result of both a reduced glycolytic activity in neurons associated with beta-amyloid plaques and upregulation in glycolytic activity in reactive astrocytes surrounding the plaques.